Welcome to the fourth and last video for Chapter 4. Here we're going to be looking at the cytoskeleton of the cell, as well as how animal and plant cells are connected between the cells, or intercellularly. So here we have a cell on the right, and I see there are so many things inside this eukaryotic cell. What if I take the cell and I start plucking out all of the organelles? I pull out the nucleus, the endoplasmic reticula, pull out all the mitochondria, etc. What would be left inside the cell? Would it be nothing? No, I would see in the cytoplasm there would still be a ton of things like ions, salts, calcium, the macromolecules like lipids, carbohydrates, proteins. I'll see protein fibers and something called the cytoskeleton. So the cytoskeleton are a set of protein fibers with various functions, and these include keeping the shape of the cell, holding things in place so that if I were to pick up the cell and shake it like a salt shaker, nothing would really move around that much. It might vibrate in place a little bit. It also, or the cytoskeleton I should say, will also be involved in moving the cytoplasm so that the cell can move forward or backwards in some cases, or to allow the transport of certain organelles or structures from one side of the cell all the way to the other side of the cell. There are three major components of the cytoskeleton, and they differ in their size as well as their function, although sometimes their functions slightly overlap. Here we have the three components listed in order of size, with microfilaments being the smallest. So I see those here. Intermediate filaments are moderate in size, and then microtubules are the largest. So very briefly, what do? Microfilaments are often involved in the shape of the cell. So certain types of organisms called amoeba or amoebas, they have these extensions in their cell membrane, and the extensions are formed through changing, breaking down, and rebuilding of these microfilaments. Intermediate filaments, Intermediate filaments are really good at stability, cell stability. And if I imagine this were a cell, they're really good at keeping things in place, holding the organelles and structures in place within the cell. And finally, our largest microtubules are very good at movement, being involved in movement. They could act as train tracks, for example, in the cell, where if I wanted to move an organelle or a vesicle from this side of the cell to the other end of the cell, the vesicle could attach itself to the microtubule and run across the microtubule's length to the other side. We're going to see later on microtubules are also involved in the movement of chromosomes, or our DNA, during cell division, and they're also found in structures called flagella and cilia, we'll see in a few minutes, and these are also involved in movement of the cell. So let's go through each of these in detail, starting with the smallest or narrowest one. Microfilaments are made from actin subunits. So each of these one single green sphere-like structures is an actin protein subunit. You have two chains of them that are intertwined to produce your microfilaments. As I mentioned earlier, these are primarily involved in cell shape. But by changing the shape of the cell through breaking down and rebuilding of these microfilaments, it can allow the cell to move, or even just part of the cell to move. So these actin subunits can easily break down and reform pretty quickly, allowing these microfilaments to change shape and change the shape of the cell. Our second component are our intermediate filaments. They're intermediate in size as well. And remember, they're not really involved in cell movement. Their role is really cell stability and maintaining the structure of the cell. They hold things in place like our organelles. These can often be comprised of pretty stable proteins such as keratin. Keratin is the same kind of protein that makes up our hair and nails. So these are very good at maintaining structure and again, stability within the cell. And our largest cytoskeletal component are the microtubules. These, kind of like how they sound, tubules, these are actually hollow tubes that are composed of tubulin dimers. So here, if I look closely at this picture, I can see each dimer is made of an alpha subunit and a beta subunit connected together. If I look down the tube of the microtubule, I can see that there are actually 13 dimers looking down into the tube. And this gives us a nice look from the top. I can see there are 13 polymerized dimers, and the dimers would be aligned up and down. You can't really see from this picture. And each of these makes up one of these tubes shown in the little cross section here. These are again involved in movement of structures within the cell, and we're going to see these structures include things like DNA during the chromosomal movement that happens during cell division, or even moving large structures within the cell. These are also going to be making up, microtubules make up our flagella, cilia, and centrioles. We'll see more in a bit. Since cilia and flagella are involved in movement, it makes sense that they're made up of microtubules. So in both, if you were to take a cross-section of a flagellum or one of these tiny proteins over here, a cilium, then what you would see is looking down into the flagellum or cilium, you would see this 9+2 arrangement of microtubules, where there are 9 doublets of microtubules on the outside, and 2 unfused microtubules in the center. There are also spokes, but it's hard to see, but spokes connect the doublets on the inside to that outer ring. Flagella are found again on the outside of some organisms or some cells like bacteria, and usually you only see one or a few on the outside of the cell at one end of the cell. Cilia are different because they're much shorter than flagella, and they usually surround the entire surface of the cell or organism. Again, both of them are involved in movement, and sometimes my students will ask me, wait a second, this picture tells me that this organism is moving to the right. Is that wrong? And that's not always wrong. It looks like it might go this way, but in this case, it uses it to pull the organism to the right. And cilia, they tend to move a little bit differently. They have this beat or wave-like motion that can allow the cell to move, that can propel the cell, or in some cases, it allows us to move things across the surface of the cell. And this is true for something like our upper respiratory system. There might be debris in our trachea, the tube that leads to the lungs, and we can have it attached to the cilia and the mucus that's found on the cell surface, and our cilia can build or propel the movement back up, so we can either swallow or spit out whatever substances we've accidentally inhaled. I wanted to point out that Chapter 4 in your textbook also includes a great table that includes the major cell components that are covered in Chapter 4 and tells us if they're present in prokaryotes like bacteria or not, and also if they're present in eukaryotic cells, and also more specifically, whether they're found in animal or plant cells or in both. And this slide is a continuation of that table. I couldn't fit the whole table onto one slide while being able to read the words. But for this second part of the table, I wanted to point out that although prokaryotes do not have chloroplasts because they do not have membrane-bound organelles, some of them, can still undergo photosynthesis. So just because a prokaryotic cell does not have chloroplasts, it doesn't mean that it doesn't have the other structures necessary to photosynthesize. So some bacteria, for example, like cyanobacteria, have chlorophyll and other photosynthetic pigments within its own cell membrane, allowing it to go through photosynthesis. Also, I wanted to point out that even though prokaryotes have a cytoskeleton, its cytoskeleton or their cytoskeleton is made of different types of proteins. We don't see microtubules or microfilaments or intermediate filaments in prokaryotes. Finally, there looks like there is an error in this table, as cilia are not found in prokaryotes. So cilia are found in eukaryotic cells, for example, animal cells like us, but it's not found in prokaryotes. So please remove this portion or cross it out in your notes. So we've talked all about the cell, the plasma membrane, and all of the internal components of the cell, like the different organelles. But what if we look at the outside of the cell? It's not actually just empty space or water. There are lots of different things found outside of the cell as well. And we call one major component the extracellular matrix. Extracellular meaning we're referring to outside of the cell. If we were to look outside of the cell in animals, we would see it's a jelly-like or gel-like layer. And there are three primary components outside of the cell in this extracellular matrix, which is sometimes abbreviated ECM, extracellular matrix. We have different types of proteins, such as collagen and other fibrous proteins, such as that shown here. We have glycoproteins. This is a sugar protein complex that we call proteoglycans, almost the same thing, switched in an opposite order, protein-sugar complex. And then we have proteins that link them together, kind of connecting everything together. Interestingly, this extracellular matrix can impact the genes that are inside the cell by turning them on and off if something changes the shape of the extracellular matrix. So often what happens is that some kind of signaling molecule will bind. Let's pretend this square is a signaling molecule. Some kind of signaling molecule can bind to and change the shape of the extracellular matrix. If these extracellular matrices, if these proteins are attached to receptor proteins found on the cell membrane, that can change the shape of the receptor and cause a series of signals to be sent inside of the cell through the cytoplasm and turn on or off certain genes. Finally, in the last part of Chapter 4, we're going to look at intercellular junctions. And these are junctions or connections between the cells. So the connections are there either to hold cells together, to allow cells to communicate with each other, or to prevent things from passing in between cells by accident. And the types of junctions we have really differ between plants versus animal cells. In plants, we have junctions known as plasmodesmata, and in animal cells, we have three major types: tight junctions, desmosomes, and gap junctions. And we'll look at each of these in the next few slides. The first of those junctions we're going to look at are the plasmodesmata that are found in plant cells. So this is plural, plasmodesmata. If it's singular, it's just plasmodesma singular. So we know that plant cells have cell walls surrounding their cell membrane. Luckily, in order for plant cells to communicate with one another, there's actually an opening, and this opening is the plasmodesma. This allows the cytoplasm of one cell to communicate with the cytoplasm of the next cell, or the adjacent cell. So plasmodesma allow communication between plant cells. Moving on to the types of junctions found in animal cells, the first type we're going to look at are tight junctions. So tight junctions are going to prevent things from passing between cells. And these are important, especially in the epithelial cells, so the outermost layer of cells that line our internal organs and our body cavities. So let's take, for example, the intestines. So after I eat food, it goes down into my stomach and then goes into the small and then later large intestines. This could be a section of the small intestine where most of the digestion of the food occurs. And let's say that this is the inside where my food is found on the left, and the outside is where my body cavity lies. I know that while my body or my intestines are digesting my food, let's say food is traveling through this space right here, I don't want any of that stuff to accidentally leak into the rest of my body. Luckily, that does not happen because of the presence of these tight junctions. So again, tight junctions, their purpose is to create a seal where nothing can accidentally pass between cells. And if I were to sketch this, if I pretend I have one cell here, let's pretend that's one cell, and then an adjacent cell right next to it. The tight junctions would be right in between. Let me switch colors here. So the tight junctions hold the cells to each other so that nothing can pass in between the cells by accident. In order to get through, they would have to go through specific receptors to enter the cell, and the cell would have to permit something in order for it to pass through to the other side. The second type of junctions found in animal cells are desmosomes, and desmosomes are great at stabilizing cell-cell connections to make sure cells don't accidentally tear apart from one another. So these are made of cadherin proteins, and they act as like spot welds or kind of like staples holding cells together. So this is especially important in tissues that stretch, like the heart muscle or the lungs. You want to make sure two adjacent cells, if I have a cell here and another cell here, I want to make sure that they don't accidentally split apart from one another. We also see very stable proteins like our keratin that was found in those intermediate filaments. And again, these are only found in animal cells, not in plant cells. Finally, the last type of junction found in animal cells are gap junctions. And these are very similar to the plasmodesmata we saw first in those plant cells because they look like channels that allow communication between cells. And again, this communication is usually through the transportation of things like ions and nutrients or other molecules from cell to cell. That's how they talk to each other, by sending signals between the two cytoplasms. For animal cells, how do you form gap junctions? Gap junctions are formed by these small proteins called connexins, and when you have 6 of them together, you form a connexon that looks like a pore, and these pores can be open or closed. As you can see here, they have 2 states. When you have 2 sets of connexons, one from this cell membrane or embedded in that cell membrane, and another embedded in a second cell's membrane, and they both open at the same time, that is when that cell-cell communication can happen. So where do we find gap junctions in animal cells? One great example is in our heart muscle. So in the future, you might learn more details about the heart, but there are actually 4 chambers in human hearts. We have our atria, we have 2 atria, and then we have 2 ventricles. And when our heart contracts, both atria have to contract at the same time, and then both ventricles have to contract at the same time. But they're made of thousands and thousands of small cells. So how do they all contract at the same time? In order to do this, they have to have these gap junctions. So when an electrical signal spreads across the heart and tells one or a few of the cells to contract, that electrical exchange is quickly communicated between all of the cells of, for example, the atria, so that they contract as a unit. And then the same thing happens later with the ventricles. The electrical signal moves to the ventricles, the communication is transmitted between cells through the gap junctions, and then the ventricles contract. All right, that takes us to the end of Chapter 4. In our next video, we're going to be looking very briefly at some material from Chapter 22. And we're going to be looking at prokaryotes, namely bacteria and archaea. And most of this material will be covered really in the next course in the major series, but we're looking briefly at more prokaryotic cell examples.